Up. I guess the food is digesting. That's perfect. It's great. So, how are you guys feeling? Is everything is up to par? Are you enjoying yourself? Yes. Are you learning a lot? Yes. Okay, good. So before we bring up this next presenter, I just want to take a little bit of time to tell you guys how fortunate you are to be in the land of science. We always think about our research and things like that, but for the most part, it's you guys that make science. For the simple fact that this is a diverse panel of people here, and we all benefit from each other, each and every one of us. And so for that, I think we all deserve a round of applause for ourselves. Can we get that? Can we get that? I see you guys don't love yourselves. So, with that being said, our next speaker is a leading authority in rice genetics and genome editing. Dr. Michael Thomson is a professor in the Department of Soil and Crop Sciences at Texas A&M University, where he holds the H.M. Beechel Rice Chair. With a PhD from Cornell, Dr. Thomson spent a decade at the International Rice Research Institute in the Philippines, focusing on stress tolerance and high-throughput genotyping. Today, he co-leads the Texas A&M AgriLife Research Crop Genome Editing Lab and oversees the Multi-Crop Transformation Facility. To discuss his work in optimizing CRISPR-based approaches to crop improvement, please welcome Dr. Michael Thomson.
Great, thanks a lot, Leo. So it's really my pleasure to be talking, my first time to talk at the Plant Breeding Symposium, and it's fantastic to see actually a lot of mentions of genome editing already. So genome editing is mainstream, I think, now in plant breeding. So that's something that's very exciting for me, really to see. And so I'll just be kind of starting off trying to address, the theme of the conference and the symposium, the multidimensional breeding, and how do we combine genomics, data science, and biotechnology? But first, I'll introduce the research that we're doing in genome editing here at Texas A&M. And so we have two different labs, the Crop Genome Editing Lab, which is a research focus, trying to optimize different techniques and really optimize the entire pipeline for genome editing across different crops. And also the multi-crop transformation facility. So Marco, Myra, Marcella, who do an excellent job with the core facility here, who are providing a service to anyone who needs transformation, agrobacterium-mediated transformation services. And so that's what I'll be doing is starting off talking about how we're developing and trying to optimize some genome editing pipelines across many different species. So my early work is all on rice, actually. And so when I came to Texas A&M, I kind of branched out and started working in genome editing across multiple crops. And I'll mention that much of this work is also from Dr. Septoningsi's work. We're actually co-leading the Crop Genome Editing Lab. And so a lot of her students' work will be presented. And then I'll look at some applications really modifying known genes and pathways for crop improvement. So some applications for nutrition, some stress tolerance, flowering time, and many of them modifying known biochemical pathways, essentially showing how does genome editing, what is the power really to achieve the traits that we're interested in. But near the end, I kind of wanted to come back to the theme of the symposium and do a little bit of thinking, brainstorming on the thoughts of really what is the future of plant breeding, and also how does genome editing fit into that. But I wanted to have a little bit of time on allele mining, which is kind of my own kind of interest for many years, and then some potentially transformative and disruptive technologies that may come 20 years in the future that we can start thinking about now. How will that change plant breeding? So just to jump into it, I think thankfully everyone knows about CRISPR-Cas technology now. It's been mentioned in almost every talk that essentially we have this single enzyme that can essentially make a double-strand break at any targeted region in the genome. And it's really amazing that essentially that it's a single enzyme and that it can be targeted to any region. But it's not just the double-strand break, it's also what happens after that, and that's the innate DNA repair mechanism, so either non-homologous end joining or homology-directed recombination. So we take advantage of these to do all the different edits that we want. This could be knocking out a gene, inserting genes, modifying amino acids, and not shown here, but also transcriptional up and down regulation, modifying epigenetic imprinting. So there's really boundless applications for CRISPR genome editing. So we're kind of, what we'll do is focus in a bit about I'll start off with what we're doing in our lab. And so this is the crop genome editing lab that was established around 2017, 2018 with support from Texas A&M AgriLife Research and the College of Agriculture and Life Sciences. And so essentially, I joined Texas A&M in 2015 after having spent 10 years at Erie in the Philippines. And when I came, we of course realized that CRISPR was a powerful technology, and we wanted to, of course, get this set up in rice, but then really have a vision to expand across many different crops and really help kind of have a centralized facility to do optimization of techniques and protocols. So the end goal is actually to have a high throughput pipeline for genome editing across different species. And so I'll just kind of run through some of the projects that our current students are working on, just to give you a flavor of what we're kind of doing with the technology now. A lot of it, I'll start off actually with protoplast isolation, because we found this a very useful tool in the lab. And essentially, so with protoplast, you could do a very quick transformation of your gene editing reagents. So we can optimize, for instance, promoters. Which promoters do we want to use for expression? We can optimize the guide RNAs of which ones are working better, and also even completely new gene editing systems. So essentially, as you know, protoplast, you essentially just degrade the cell wall, and then you have the individual cells that you can then transfect very quickly, and then do either expression analysis or sequencing to identify the mutations in those protoplasts. So this first project was actually a student of Dr. Septi Roli, and she's doing this in wheat, actually, and so she's been successful. So of course, this first step is to optimize the protoplast isolation protocol for each species. It's quite a bit different across species. And then checking whether it works for genome editing. And so she had success in wheat with that. And also, I'll mention that, so protoplast is one technology. Another is leaf infiltration as well. And so we often do either agrobacterium or other types of leaf infiltration. I'll talk about just, again, a transient assay that's very fast that you can use as you're optimizing the process. So this has been done now in our group in many different species. And so some of these are published, so I won't spend too much time on it, but essentially looking at across like peanut, cowpea, chickpea, even guar, and these are again through collaborations with various groups, so some of them within AgriLife, so Dr. Waltram's group, and some of them internationally. So we often have visiting scholars come as well. So these are, you can see just essentially, again, protoplasts, using them as a validation tool for the system. And then more recently, actually, Deepat, so he gave an excellent talk looking at rice, but his main project is in mung bean and essentially modifying a gene, LOX2 gene that gives mung bean actually the beanie flavor, or a disc flavor, you may call it. So essentially, he's done an excellent job of optimizing the protoplast and getting that up and running in mung bean as well. So a lot of these, I'm just showing that essentially we have these tools that we can then start to optimize the system before we go into stable transformation. And so I'll talk also about some alternative delivery and transformation techniques. One of them is with nanoparticles, so carbon nanotube-based delivery. And actually this technology was developed at UC Berkeley in Marquito Landry's lab, where they identified that these single-wall carbon nanotubes, you can attach plasmids, you can attach RNA. More recently, they've attached proteins, and it actually passively diffuse through the plant cells into plant cell tissue, so through even the cell wall and the cell membranes. And so we had a project looking at, okay, can we get this up and running? We started off in rice, of course, so leaf infiltration is a lot more difficult in rice than some other species. So we're using a modified kind of infiltration method where you're actually puncturing the leaves. And then essentially the idea is that we soak it in the carbon nanotube solution that has our plasmids attached to those carbon nanotubes to see if it can actually deliver that cargo into the plant tissues and then be expressed and translated. And so that was a project with leaf infiltration. We're using GUS expression. And then more importantly, we want to essentially test approaches to do what's called implanted transformation. So this is alternative to the normal in vitro tissue culture process. So I know there's many groups around the world actually looking at how do we essentially bypass tissue culture because it is such a lengthy, kind of cumbersome process. So one way potentially to do that would be implanted transformation. So essentially taking either the seeds or, so in Arabidopsis, you can do floral dipping, right? So you can dip it into the agrobacterium, but for most crops, that's not gonna work. It's been tested and it hasn't been reproduced. So we're trying to find alternative methods for sort of that kind of implanted transformation. So the CNT delivery actually into either mature seed excised embryos or into just the imbibed seeds as they're before germination to see if we can get, again, deliver the reporter genes and then the CRISPR-Cas9 cargo to have, essentially the idea is that we introduce it at the seed and then we can just germinate them and grow them normally instead of going through, again, that lengthy tissue culture process. So this has been published, but the end result is that it's not quite as easy as it sounds. I mean, these kind of alternative techniques often are a lot more difficult in practice. And so that is true for this one as well. And so I don't see it as replacing agrobacterium-mediated transformation in tissue culture anytime soon, but it is under active investigation. So, and then that's also been tested, so that was with rice, and then Merve has been testing this also in cowpea leaves, and this was recently published in PLOS One, where both carbon nanotubes and carbon dots, and again, attaching cargo to the surface and seeing if they can infiltrate either through leaf infiltration or again, potentially testing seeds as well. But in this case, she did have successful gene editing. So attached to the carbon nanotubes and carbon dots would be the CRISPR-Cas9 expressing plasmid with the guide RNA. And so often we use, for instance, guide RNAs targeting a visible, so the knockout will give a visible phenotype such as PDS. And so that's one of the successful uses of nanoparticles for delivery. But again, it needs more optimization before it can be used in a high throughput fashion. So that brings us to our stable transformation, which is the multi-crop transformation facility here on campus. And again, this is agrobacterium-mediated transformation using in vitro tissue culture for regeneration. And of course, and we're looking for alternative techniques, but it turns out that currently this is the most robust and essentially most routine technique that we have available. And so, most of our genome editing is going to be using, agrobacterium transformation and in vitro tissue culture. But the trick here is, as many of you are familiar with transformation labs, it's more of an art along with the science. And so there's a lot of kind of parameters that you need to optimize. And so Marco, Myra, and Marcella have really done an excellent job really to optimize that type of transformation. So as you know, there's, so here's some of the crops they're working on. So for each crop, often there's a single genotype that's essentially transformation amenable. So for sorghum, it's like TX430. So one of the really challenges that they're working on is how do you optimize for additional genotypes? As was mentioned earlier, having genotype independent transformation. And then essentially, so additional genotypes, but also speeding up the process is important, and so they've been working on that as well. But of course, as a service lab, I mean, they're open to anyone who's interested to collaborate and actually make use of the lab. And so they're ready to start novel protocol development. So they've optimized at least one genotype, sometimes more for each of these crops. But they're always happy to start new crops or new genotypes as well. So they're really a fantastic facility. So I encourage everyone, if you need transformation services on campus, to actually use them. And then here's some examples of some of the work that they've done in gene editing as well. So generally what we do is when we work in partnership is my lab will be developing, like designing the guide RNA constructs, actually developing the plasmid vectors, putting them in the agrobacterium, and then passing them to the transformation facility. And then at the end of the day, when they do actually the hard work of getting those transformed and regenerated, we'll help with the molecular characterization. And so we work together on that. So they've been quite successful with some gene editing projects. So now I just wanted to kind of talk a little bit about, okay, how do we use genome editing? What applications? So what's the future hold? And I wanted to kind of frame it with the background of essentially the genetic diversity that we have available. So the crop genetic resources, the natural variation. So I'll be focusing entirely on natural variation, not really exploring synthetic biology, but essentially there's abundant natural variation for every crop that we're working on. At the same time, we have this fantastic knowledge of genomics, gene discovery, biochemical pathways, also now with resequencing efforts, alleles for each of these target genes. And with all the genomics data, we want to find a way to make that worthwhile and make it usable. So I'll be starting off by showing just some proof of concept and pilot projects of specific applications where we're using genome editing for specific traits by modifying essentially known genes and pathways. So there's quite a few examples we have in the lab. And then the second half of the talk, I'll be looking more towards the future perspectives. I do want to spend some time on allele validation, transfer, and stacking as well. We've already heard some stacking from Corteva, and then some CRISPR-driven, potentially transformative technologies that I'll end off with. So now I'll just run through, again, some projects in the lab that have targeted certain specific traits. I'll start off with rice. Those are the ones we had really started off when we first opened the lab. We immediately started some seed projects, and rice was some of the first ones. So this first one is essentially looking at, can we develop high lysine rice by modifying the biochemical pathway. So if you see, so this is lysine, and of course this has been kind of proposed earlier by using like RNAi and others. But essentially what we want to do is knock out the two genes below lysine so that it accumulates higher lysine instead of being essentially converted into other molecules. And so this was a project by Khushbu Rastogi. She was a student, again, of Dr. Septi. And she was successful, actually, in going through this whole process of CRISPR editing, knocking out the LKR SDH genes, and achieving 7.8% high lysine in the gene-edited lines. And so that was also published last year. And so another important project that we'd like to show as kind of the potential application is actually in resistant starch. So there's a lot of interest recently in high fiber and higher resistant starch, which is essentially a slower digestion. So again, I think as Deepa already introduced the glycemic index, rice has very high glycemic index. We want it to be essentially slower digest and lower that glycemic index. And so one way to do that in rice is by modifying the resistant starch levels. And so I think Deepak did an excellent job of showing, what's the ongoing work. I will mention this original work was done by our postdoc, Sudip Biswas, and he was successful to knock out, essentially we targeted all four of the starch biosynthesis genes and achieve those levels of 15% resistant starch, whereas the wild type is near zero resistant starch. So this is published a couple years back, but essentially showing, again, some of the applications that we can do. Now, I did want to highlight Leo's work as well. So he's right in the middle of his project working in cannabis. I have him on two projects, rice and cannabis. And this is, we're excited to be using the Ruby reporter gene. So we've seen this in the literature, and it's just a nice way to check our transformation if it's working, since it'll essentially producing a red pigment in your tissues. And at the same time, I've asked Leo actually to try to develop an implanted transformation procedure for cannabis as well. So of course in Texas, industrial hemp, which is approved, but essentially what we want, we still have that concern with the THC sometimes going hot for the farmers. So Leo's end goal will be, as with many others, knocking out the THC, but in this case, he wants to actually knock out all the cannabinoids, the entire pathway, so that we can have essentially zero THC instead of having that concern for farmers. So he's made some recent, some nice progress, really just starting to test. As you may or may not know, actually cannabis is extremely recalcitrant to transformation and regeneration. There's very few labs that can do it. I think Wisconsin's one of the ones that has done a great job, as Larry mentioned. But so we're trying to get that transformation process optimized before we move on to some of the applications. Okay, a couple more examples. One is we do a lot of work with mapping QTLs or finding GWAS hits and wanting to essentially validate the candidate genes underlying these QTL, find map QTLs or GWAS significant SNP associations. And so one of these is with anaerobic germination. So again, Dr. Septi is working on flooding tolerance. And so they've identified a major QTL in chromosome 6, and I've identified several candidate genes. Now they're overexpressing and knocking out these genes in 2 different rice varieties. So this is just a powerful technique there. Genome editing really does help us with that last step. So fine mapping, you may get a handful of genes, but you don't know which one's the causal variant until you do the actual validation. And so either knocking out or overexpressing or doing both is very powerful. And then next, sorry. So again, we have a project working on stress tolerance, and many of these are in rice because that's mostly our background, but essentially looking at high nighttime temperature tolerance in rice. And this is a project in collaboration with the AgriLife Center in Beaumont with Dr. Lee Tarpley, working on specifically high nighttime temperature, because in rice, high temperatures cause a severe decrease in the grain quality as well as potentially in the yield. And it's not just high temperatures in the day, it's actually the high nighttime temperatures that have that significant effect. And so this project funded by FFAR has looked at a few, again, so genes that we think are important for high nighttime temperature tolerance, and actually going in and overexpressing and knocking out these genes. And you can see, and these are the overexpressed lines compared to the wild type Presidio and then Chiherong, that it's a bit harder to see here, but that they are actually better performing under high temperatures. But this is essentially still in progress with Rolly as Septi's graduate student. And then looking at, there's heat stress in cowpea. So this is a project collaboration with Hangbin Zhang as well in our department, where Merve has been working to, again, validate major genes underlying important, in this case, QTL for heat stress tolerance. And the idea is that, okay, breeders can use the QTL with flanking markers, but they can only do that for cowpea. So if we want to identify, okay, what are the major genes controlling heat stress tolerance in cowpea that we want to then see, can we use those genes in other species? You really do need to clone the gene and identify the causal variant. And so that's an important exercise to isolate the causal gene for that. And so this is a very valuable target that we're right in the middle of working. So Merve is essentially designing these, in this case, the overexpression plasmids, and then the knockout plasmids as well, to knock out. So they have 2 candidate genes for this fine map region, and then using overexpression, and then for the knockout using CRISPR combo, which is, I'll talk a little bit more later, but essentially a way to have a more efficient transformation approach at the same time as knocking out a gene. But I'll come back to that part. So, and as well, so another project Sudip Biswas is working on, in this case, heat tolerance in rice, again, CRISPR combo, but in this case, it's CRISPR combo with base editing. So we can use that approach to have more efficient transformation with base editing or with knocking out a gene. So he's essentially identified, he's successful to have 15 plants that had the base edit for this particular heat tolerance gene. And this is some major genes that are known for heat tolerance, but the specific causal variant is essentially we're just confirming that functional nucleotide polymorphism that that has been proposed to actually control heat tolerance with this gene. Okay, so then that brings me to another project, which is actually flowering time. So this is one that I've been working on for quite some time. And if you look at flowering time in Arabidopsis, it's very well known, the pathways, and then in rice as well. So there's short day and long day flowering time pathways. And essentially we know which ones are activators, and so like the florogen genes, these FT-like genes, and then which ones are essentially the repressors of these flowering time pathways. And so I had a student, Karina Morales, who knocked out, for instance, the activators, and essentially you get plants that never flower. And then on the other side of the spectrum, we want to actually knock out some of the repressors to see if we can kind of fine tune early flowering using genome editing as an approach. So we started off rather ambitiously by actually doing multiplex knockouts of 6 genes at the same time. It turns out that that's quite complicated when you're in practice, when you're trying to analyze all of these genes. But essentially right here, you can see the phenotype. So this is, for instance, the T1 progeny in the greenhouse. So if you haven't seen it, but in rice, the flowers will actually extrude the anthers during the flowering so that we can identify, okay, essentially days to flowering for rice, which again, that's our trait of interest. So with the 6 genes, I've had 2 more students actually working on this. So I had Ashifa, a master's student who's now graduated, and then it was passed to Leo actually to wrap this project up. And so with quite a complicated project, so with 6 genes edited at the same time, you don't necessarily get knockouts in all the subsequent progeny. So we actually get combinations of knockouts. And so we're getting, double mutants, triple mutants, quadruple mutants, all of them, potentially affecting flowering time in different ways. And so essentially now we have like the T2 seeds. We did see some potentially promising early flowering lines at the T1 stage, but we need to confirm that at T2. And so Leo will be working on that to see if we can identify what will provide early flowering in that project. So all of that was just really an introduction to the lab and some of really what ways we've been using genome editing right now and using current state-of-the-art techniques. So I did want to kind of talk about a little bit about, okay, looking in the future, what are we seeing as potentially promising technologies that we can move into. And I did want to spend some time on this concept of allele mining. And so this is something that's really stuck with me since my time at Erie. They do really an excellent job with this, and I think there is untapped potential. And what we're talking about is when you see the publications of major genes that have been cloned that will increase the yield of rice for 20%, or major Nature science papers that talk about these important genes, what we see is that very few of them have actually made an impact in plant breeding. And so often they'll get the publication and then it'll be essentially forgotten for various reasons. And so how do we leverage not just the clone major genes in the top publications, but actually all of the genomics data that we have available to us? So I think if you look at like the NSF Plant Genome Program, for several decades, they've made these investments in genomics for crop plants. And really the question is, what kind of impact has that had, and how can we actually leverage all that data now moving forward? So I think the impact has been, I would say, rather untapped compared to what it can be. And I'll kind of frame this in the bigger picture of really starting with large effect rare alleles. And this comes partially from my background in rice, because rice as a species has many large effect major genes and alleles, which is quite different from other, for instance, from maize, that's more, quantitative, hundreds or thousands of minor effect genes. But essentially, I think some of the principles can apply across species. So I think really I want to start off with this idea that more work can be needed to test high value large effect genes, and specifically exotic alleles. And so there's a lot of really untapped genetic diversity around the world in gene banks, in CG centers and national programs that really potentially could add novel traits. They can add, resistance to emerging pests and diseases. But if you take, for instance, your genomic selection program, you don't want to be directly introducing exotic germplasm. It'll essentially destroy your population. It'll bring down, everything you've worked hard to achieve. And so the first challenge is thinking about, okay, how do we introduce exotic germplasm into a very successful active genomic selection program, or phenomic selection as it may be. So essentially the idea here is that we want to find a way to more efficiently use that exotic germplasm. And so, of course, the first step is you need to know where are the valuable alleles. A lot of this has been done already. There's thousands of QTL studies, GWAS publications. Again, a lot of it not having the impact that it should have. So I think a lot more can be done to really kind of mine that data and identifying which essentially genes that are the most promising that we can then move forward. And currently, the way people generally do that is you take that high value allele and you put it in the background of a, say, elite genetic background, so a near genetic line. And we'll kind of talk about the limitations of that. But first, I think one kind of example of why these rare large effect alleles are actually worth the effort. There's a lot of work done at Erie to actually look at their gene bank. They have 100,000 rice successions. What are the most valuable ones, and where are the rare alleles that are most useful? And so something like anaerobic germination. So this is a trait in rice where essentially, 99% of the rice lines will not be able to germinate under like 10 centimeters of water for like 10 days because they're completely submerged. And so there's this very rare trait that you can find in literally less than 0.06% of the germplasm. So they identified, after screening like 8,000 lines, they only identified 19 that actually had survival under these anaerobic germination conditions. And so that's the point here, is that those are essentially large effect and then rare alleles. And then this idea of QTIL essentially, isolating in near-isogenic lines and then pyramiding them has been around for several decades as well. So this is just an example. For instance, again, in rice, you may have multiple loci for each trait. You can develop near-isogenic lines. If these are from exotic germplasm, again, you don't want the negative linkage drag. So you want to be able to combine those. And so one way would be to develop near-isogenic lines of all of these high-value targets in the same genetic background. And then you could just intercross them at the end of the day. And this has been done several times at Erie and in other places, but it's a huge amount of work for a relatively small number of genes. And so we'll talk about, maybe there's some ways to do this more efficiently. And kind of on a side note, this type of work is still ongoing, and thankfully we do have some collaborations globally here at Texas A&M where we're doing this type of allele discovery and allele transfer. And I did want to just highlight this one particular project, and it's kind of exciting because this is exciting and unfortunate, but it is one of the few projects that survived the USAID shutdown, where essentially USAID's most of the portfolio was shut down, but some of it was saved and transferred to the State Department. And so this is one of the few programs that is continuing at the U.S. Department of State. So we're very grateful that they have continued some of these projects. But essentially, it's the Climate Resilient Cereals Innovation Lab. So this is actually led by Kansas State University. But it's in collaboration, it's connecting U.S. universities with international partners to do allele discovery and allele transfer. And so our particular partner is the Bangladesh Rice Research Institute in Bangladesh. And essentially we have here at Texas A&M 2 sub-awards from Kansas State to really explore allele discovery with our partners. And so one is my project on heat tolerance, and then the second is with Dr. Septanisi on flooding tolerance. And so this is essentially following this idea of identifying, again, these major large effect genes, transferring them to elite genetic backgrounds to validate them, and then essentially introducing them into breeding pipelines. And so I'll just show real briefly, this is actually just started, this project. So I'm working with 2 scientists in Bangladesh on looking at their Aus germplasm. So those of you who aren't familiar with rice, there's actually 5 major subgroups. And if you look at indica and japonica as kind of super subgroups, within indica you have both your normal indica and then you have Aus. So Aus are essentially specific rice accessions from Bangladesh and then parts of East India that have really a lot of unique characteristics. So many of the stress tolerances, for instance, the flooding tolerance, the sub-1 gene, and many others are from either indica or specifically Aus backgrounds. But it's largely a genetic, essentially, a genetic pool that's been largely untapped. So there's not as much research on looking at across all of the Aus accessions specifically. And so at Beery, they've assembled 346 Aus accessions, and we're specifically looking at heat tolerance. So they're heat screening them in both kind of hotspot field environments as well as heat screening facilities at Beery. And so this is, and then they'll be sending tissue over, we'll be doing the sequencing and doing the GWAS analysis with them. So I just wanted to highlight that thankfully those kinds of international partnerships are still ongoing, but hopefully there'll be even more in the future. So I think that type of partnership is extremely valuable. So I've kind of put together this kind of framework. If you're just looking at, okay, developing, near isogenic lines for very high value targets, integrating all the data we have, and then essentially getting them into an elite genetic background so there's no negative linkage drag, and then testing them, validating them across environments and conditions, and then finally deploying them to breeders, that's actually a huge amount of work. And it's very valuable work, but it would take like multiple teams to do, for instance, identifying the alleles, but also getting them into elite backgrounds very cleanly. And trying to reduce that negative linkage drag is the most challenging aspect, because inevitably you'll have a large chromosome segment. You try to reduce it, and it's quite difficult. So now it kind of brings it to the question of what are the potential, transformative or even disruptive technologies for the next 20 years. And you'll notice, so this is the vision of the future from ChatGPT. Actually, I've asked it to make an image of the future agricultural production. So of course, it's all drones and autonomous tractors and some unusual looking cornfields. But essentially, I think we can take from that, of course, we'll have precision phenotyping. It'll be largely autonomous. We already have large-scale, low-cost genotyping. So I think that efficient genomic selection processes, as aided by AI in crop modeling, predictive modeling, that will continue and I think accelerate. So we should see steady, genetic gains for yield continuing for many years in the future. But the question comes, what about the novel traits that may not, and rare alleles that may not exist in your elite germplasm pools? How are we introducing novel genetic variation? And essentially that would be for abiotic stresses, novel environments that the crop hasn't seen before, emerging pests and diseases, maybe nutrition traits. So what kind of, how can we use the current technologies to affect plant breeding 20 years from now? And so I wanted to kind of run through a few of these, and in case some of them I'm sure you've heard about, others may not, but I just kind of want to quickly run through the technologies. The first is the CRISPR combo I mentioned earlier, and this kind of builds upon a lot of the pioneering work at Pioneer, right? So with Baby Boom and Wuschel and other genes, and essentially taking that to improve the transformation efficiencies at the same time of using that for CRISPR. And so that's something that I think will improve over time, and not just using morphogenic regulatory genes, but there's, like I mentioned, the implanter approaches as well. So all of that I think will continue to make CRISPR gene editing more efficient. Next, there's this idea of developing essentially de novo domestication. So if you haven't heard about it, essentially using, again, CRISPR to edit all the known domestication genes, but editing in the wild species to essentially rapidly domesticate it to become, essentially to have the traits that, the domestication process has selected for many generations, but doing that in essentially a single experiment. So this is kind of ongoing work by Cornell and Cold Spring Harbor and others for testing de novo domestication. Another one that's kind of hit recent publications is apomixis, and so this has been a long-term goal, and now with CRISPR technologies, actually they've already achieved some amount of apomixis where you can fix hybrid vigor and continue that moving forward. Of course, it's very complicated and transgenic, and so it has its issues, but essentially looking at in the future, it could be a potential again, disruptive technology. But one of the ones I want to focus on a little bit more is actually targeted recombination. So this is an idea where you have, right now recombination is random. So we know that in genetics, it's random recombination. The idea is, can we use CRISPR technology to target those recombinational breakpoints at the positions along the chromosome that we want, essentially have targeted recombination. And really the value of this, of course, is if you have, genomic estimated breeding values along a chromosome, you actually can envision your ideal kind of idiotype of what are the best chromosome segments, but you're relying on natural or random recombination. So you need very large populations, multiple cycles to continue to improve through genomic selection approach. But if you could, and this is, there is one publication and a few others that have started to actually examine, can we have targeted recombination? And so the technology is at a very early stage. I don't know if this will be, how long it'll take to become routine. But if you think about it, it is quite transformative, because then you can actually, instead of growing up large populations, you could just say, okay, I want a recombination breakpoint here, here, and here. And actually in one generation you get, or a few generations, you get kind of your ideal optimal haplotype. So that you could call that haplotype stacking. I think it's stacking is a little bit, we'll talk about, there's several definitions of that, or just optimal genotypes along your chromosome. So that if you think about it, and this is also, some ideas have been published, where you have gene pyramiding, so you have your ultimate genotype of major large effect genes, but you can also pyramid your most beneficial chromosome segments across the entire genome, essentially, without knowing which genes, specific genes you're targeting. Kind of related to this is there's a publication of essentially rapid trait stacking through speed breeding using marker-assisted selection, using backcrossing, intercrossing. And I kind of show this is that this is the kind of the pyramiding, the stacking approach done conventionally through speed breeding, but this could also be essentially accelerated both with targeted recombination, but also, as was mentioned at Corteva, actually literally just stacking them, taking genes and stacking them where you want them. And so I did want to mention, I guess with CRISPR, one is allele validation. So currently, NIL validation is extremely lengthy and time-consuming. We can use CRISPR both for validating the functional nucleotide polymorphisms, validating which ones are the causal variant genes, and then hopefully in the future, actually moving to technology that can actually just replace the alleles essentially at will. So if you have an exotic allele in this line and you want to test it in your elite genetic background, hopefully it won't be long before CRISPR matures to the point where we can do rapid allele replacements. And so this is an example of rice. We have a number of major genes, large effect genes for many different traits. All of them have been cloned, so we know that that's an important gene, but not necessarily all the alleles haven't really been characterized. So there's not as much known about the functional nucleotide polymorphisms, about which alleles actually provide the desired effect. So part of gene editing is validating the research part, and then once they're validated, how do we deploy that in breeding programs? And I think we may start to be able to do allele replacements where we get to the point where we don't have to go through the multiple backcrosses for NIL development, that we can actually do that. We might call it instant NILs, essentially. So we can take exotic germplasm. Of course, we need to know the genes and alleles to start with, so that's a fair amount of effort. But at the same time, then we can potentially just replace the allele. And I do use the term allele replacement because I think there's some value in having, if you know that you have, for instance, a susceptible allele in your germplasm, you want the tolerant allele in the exact same location. There's some value in having that precise replacement, but at the same time, I think there's a lot of value in just like an allele stacking where you just, let's say you want them all in the same locus, and like Corteva, the disease super locus was absolutely fantastic, and so I think that's really the cutting edge of what we'll see in the future. And so this is kind of a related approach, but more on the research focus of, okay, we want, say, to test 6 different alleles for this locus. We can make an allelic series where we actually replace and have all 6 combinations to see what's the phenotype. So in the end, just wrapping up, I think we'll hopefully have this vision of, for quantitative variation, have the targeted recombination providing this haplotype optimization much faster than current genomic selection, while at the same time, allele replacements providing this exotic allele deployment. And it would be nice to combine that with a rapid generation advanced speed breeding approach and really bring it down to almost an engineering pipeline of design, build, and test. So you're designing a genomic design of these optimal haplotypes and exotic alleles. You're building essentially those haplotype combinations using these technologies and then rapidly testing them under your multiple environments, under your precision phenotyping, and then releasing and deploying those across target environments. So hopefully that kind of is a little thought-provoking on what the future may hold. I think there's a lot of promise now of what CRISPR can do. Currently, most people are doing knockouts, but I think in the future we'll really extend that to a lot more applications. And I didn't go into all of course, we still have to deal with regulatory issues, IP, consumer acceptance, but hopefully that will also improve in the future. So just to acknowledge my lab, many of them actually helping with the Plant Breeding Symposium this year, and also the funding from USDA NIFA, FFAR, the IHA here at Texas A&M, and also US Department of State, AgriLife Research, and Texas A&M. And I do want to also plug the NAPB meeting. So we're very excited to be hosting NAPB here in College Station in June, so June 15 and 18. And in particular, the pre-conference workshops. I'm actually helping organize on June 14 many of these pre-conference workshops. So I do encourage everyone to go ahead. The registration is live online. online, go ahead and find the website and look at that. And I think we'll have a really amazing time at NAPB in June. So with that, thank you, and I'll answer any questions. Yeah, here, T.
Mike, very good presentation and certainly great ideas looking into the future as to what CRISPR can do. But the limitation still is putting all these components, CRISPR components, into the cell, especially for gene replacement or insertion. We're still not there yet. And I think rice would be a good candidate because you can isolate protoplast from rice and you can get the same protoplast to go back into the whole plant. It's very hard with other monocots as far as I know, unless Corteva is better at doing that.
Yeah, that's an excellent point. And I think if we did have efficient protoplast regeneration, there's a lot you can do with protoplast, like in a high throughput manner, to test hundreds of thousands of combinations and then potentially pick the protoplast that you want and then regenerate. That would be the ultimate solution, but I think we're not there yet. I agree that's why I mentioned it's 20 years down the road. So hopefully the technologies will mature, but that's my out we're looking in the future. So not current technologies. So.
Thanks.
Yeah, one more in the back.
Okay, you have given quite a few examples. Let me just focus on the one where you have 7.8% lysine in the rice. So after the transformation, how about its other agronomic traits like yield, aroma? Did it have any adverse effect?
Yeah, so we've done very preliminary experiments just in the greenhouse, and we haven't seen, and we've looked specifically at the edited versus wild type, and we haven't seen anything obvious at the greenhouse level. But of course, all of these would have to be, validated, at the field level eventually if we're going to think about like commercializing and things. But right now it's a proof of concept.
It's just inside the greenhouse.
But with greenhouse data, they don't have any negative effect. Okay. And the same with the starch branching as well.
But field data is still to come.
That's right. Yeah, we haven't done actually any field testing on any of these lines. So.
Excellent talk. Thank you so much for all that you've done for the symposium this year. You clearly have a lab culture that generates exemplary graduate students. So what are some of the elements of lab culture that you've developed and cultivated to make such excellent graduate students?
Well, and actually, it's all on them, actually, because the culture comes from the students themselves, and they are actually very helpful to each other. And so it's nothing I do, actually. I'm more of a hands-off supervisor. Actually, I let them do their thing, and then they find out that, well, I'm not going to help them. So they ask each other, and then actually they built this culture of helping each other, which I think is fantastic. So that's, in some ways, you have to let them figure it out. So I think that's where they get it from. So
please, let's think. Oh, yeah. I'm 0 for 2 today, guys. I'm so sorry. We've got an additional question.
I will not ask the other questions. Very impressive talk. You have worked on many crops, most of the first crop. Do you have any plan to work on the tree?
Tree crops?
For example, the pecan. I work on the pecan.
Oh, right.
The pecan doesn't have any transformation system available right now. So how do you, if possible, to work on the tree?
Yeah. So, I mean, that would be something you can ask, for instance, Marco and Myra in the transformation lab. They have checked, coffee, I believe, and maybe roses a little bit. But you can talk to them because they're the experts in transformation. But I agree, like, we definitely need the transformation optimized first before you can start applying these technologies.
Thank you.
Okay, thanks.